students, now let us see few important terms in biostatistics. Before we start the collection of data, we need to understand some important terms which are used in the biostatistic paper. Now, biostatistics is the science of collection and analysis and interpretation of facts and numbers connected with biology. So, statistics which is used in biology is called biostatistics. Statistical data, its collection, its analysis, and how to interpret that data which is collected. So, all these things are studied in biostatistics. Okay, so the first important term is data. The values recorded in an experiment or observation are called data. So, what is data? Data means whatever values we record in our experiment or observation is called data. If you have single data, it is also known as data, and if you have more number of data with you, then it is also called data. That means for plural and singular, the term same data is used. Okay, then the next point, that is there are types of data, that mean those are primary and secondary data. So first type of data is primary data. So the data which is collected by investigator is known as primary data, and secondary data is the data collected by another source, example by newspaper, from books, or any journal or any other agency. If you are collecting that data from such sources, then it is known as secondary data. Okay, so in short, the primary data is collected directly or by the investigator himself, and the secondary data is the data which is collected by another source if the investigator is not himself getting that data. Okay, so this is by another source, like any newspaper. If the books are having some data and you are collecting that data from the book, if the journal or any other agency, if you are collecting all the data information, any type of information you are collecting from any other sources, not directly, then it is known as secondary data. Then the next is samples. The next term is sample. The small representative fraction of a population is called a sample. Getting a sample from a population is called sampling. Okay, so see the example, then you will understand what exactly the sample is. Only rice grains, some or few rice grains are checked from a boiling pot. When we boil the rice, or we cook the rice in any container, what we do? We just collect some of the grains from that pot, boiling pot, and we check it whether the rice is cooked or not. Okay, means we take a small sample from the whole rice which is boiling in the pot. So that is known as sample. A small representative fraction of a population is called a sample. Then if you take any other example, like if you buy any fruits and vegetable, what we do? We just test one of the grape or one of the piece of that fruit. You ask the shopkeeper or the farmer to get a small piece of that fruit, and you test it, and then you check that whether you want to buy it or not. You decide it after sampling or by testing. So it is called a sampling method or the sample. Okay, this is the second term. Then the third term is variable. The value of an item or individual is called variable. Example, height of student is a variable. Number of mango trees in a garden is a variable. So the value of an item or individual is called variable. Any value, any number, whatever data you are studying, that number is your variable, right? The next is types of variable. There are two types of variables. One is qualitative and quantitative. Quantitative variable is When a variable is measurable, it is called measurable, it is called a measurable variable. And the example is the number of students or the height of the students, weight of the students. So whatever data you are getting, that can be measurable, and in that case we can say it is a quantitative variable. Quantity is there in that variable. Okay, so this is the first type, that is quantitative variable. And the qualitative is the second type, in which the quality of that variable or quality of that product is taken. So like for example, color of flower, color of skin, all these are unmeasurable, these are not measured. Okay, just we by just observing that variable, we have to collect that data. So this is not much common in our statistical analysis or stat study. So the first one is called quantitative variable, which is frequently or very commonly studied. Quantitative variable is again of two types, namely discontinuous and second is continuous. Discontinuous variables are in whole numbers. Okay, so discontinuous variables are in whole numbers, whereas continuous are in decimal numbers. They are continuous, so we get those values in decimal points. And continuous variables are the numbers 1, 2, 3, 4 or 25 and 92. So all these are continuous variables, whereas if the data or if that variable is in number, but those numbers are in decimal points, like 1.5 is the height of the particular animal, or 1.8 or 2.5, anything. So all these numbers are in decimal points or decimal numbers. This type of data, this type of variable is called as discontinuous variable. Now let us understand the collection of data, how the data is collected. Those methods are given in the next part, but before that we need to understand what is collection of data and why it is important. The process of getting values and facts from an observation or experiment is called collection of data. The first one is primary data. Primary data is one which is collected by investigator himself for the first time. The person collecting the data is called an investigator. So as I told just now, that the primary data is the data which is collected by an investigator himself for the first time. This is a pure data or original data, we can say. The person who is collecting that data is called investigator. Okay, and what are the features of this primary data? It is first hand information. It is first hand information, means the investigator is collecting that data, means it is not the data which is collected by any other person or any other agency. So directly you or any person who is collecting that data, and that data is called as primary data. Next, it is original in nature. There is very less original in nature, and then it is accurate and more reliable. The data which is collected by the investigator is obviously it must be accurate and reliable, but it consumes more money and time. That is one of the drawback or drawback of this method is the first primary data takes more money, or it needs more money, because the investigator has to collect the data from the from some experiment or from some other study. So all these things needs money, and therefore this is one of the disorder or drawback of this primary data collection. But the plus points are, it is the originality of this data, then accuracy, reliable, it is all these are the positive things, or we can say these are these all are positive things about the primary data. Okay. Now one more example is given, like the amount of oxygen in water sample by the student is primary data. So one example is given, that is the amount of oxygen in water sample by the student. The student is collecting the sample, and that from that sample he is calculating, estimating oxygen amount of that water sample, and the data which is collected, the information which is written is the primary data. And if the secondary data is there, in that case it is the one which has been collected by a source other than investigator. So suppose if the primary data is the experiment done by yourself, okay, you are doing experiment in the laboratory and you are taking all the samples and you are calculating the oxygen which is present in that sample, and you are writing all those things in your notebook, right? So that data is primary data. And if your friend or somebody else is taking that data from you for his own experiment, or just he is copying all those things in his notebook, then that data which is collected by him, your friend, that is secondary data, right? So these are the two things which is to be understood. Then the secondary data, it takes less time. How? Because the person who is collecting that data, just now I given you an example that if your friend is collecting the data from your study, your experiment, you have done it, you have to spend time for it. But when your friend is collecting that data from your book, it obviously it will take very less time. So this takes very less time. It cannot be original. If you are manipulating those numbers while giving or before giving it to other person, then it may not be original in that time, isn't it? Then relatively less costly or less time it will require. It is less reliable. So these are the things which are for the secondary data. Then sources of secondary data. Sources are the data which can be collected from published sources or unpublished sources. Like for example, if you are taking published sources, then the examples are data in magazine, periodicals, newspapers or books. If you are taking that data information from any kind of magazines or periodicals or newspapers or books, that is published type of sources, right? This is one of the source. And second source is unpublished sources, unpublished data, like records maintained by a various government and private offices. This is the unpublished sources. If you are taking that information or data from such things like the whatever record, any record which is maintained by government and private offices. Okay. So this is all about the first point, that is collection of data, and in the next lecture we will see how the data is collected, which are the important methods to collect the data, primary data. Okay. Thank you.